Testing. Testing mic. Testing 1, 2, 3. Okay, so I think got sounds. My mic is open. Okay. Okay, so now already 8 o'clock, 8:01. So okay, so without further ado, now we will start about the class. So for today, the class is about the chapter 2. We will enter chapter 2. Yesterday we just finished chapter 1, right? So today we will enter chapter 2, which is cell structure and organelles. Okay, so about chapter 2, I think got 3 topics, right? First topic is prokaryotic and eukaryotic. Second topic is cell components. And then third topic is specialized cells. Okay, so actually chapter 2, in overall, actually you already learned in SPM lah. Almost all are learned in SPM already. But normally students, biology students are not focused at all. Even me, I as biology student, SPM pun tak fokus because this chapter is very boring because you already learn. But unfortunately, MPM in recent years lebih suka keluar chapter ni dalam soalan esei ataupun soalan struktur. So if you want to score biology, you should score lah. You should study this chapter as well. So without further ado, let us begin first topic. So for today, we will only proceed 2 topics, prokaryotic and eukaryotic, and also cell components. So prokaryotic and eukaryotic. Okay, so next. Okay, so before that, we need to learn about the cell concepts. So what is cell? Apa itu cell? Kenapa ada cell? So who proposed the cell concept, cell theory is proposed by these 2 scientists, Matthias Skandian and also Theodore Skan, I don't know how to spell, Theodore Skanwan. These 2 scientists these 2 scientists proposed the cell theory, and it proposed that all of the animals and plants, actually not only animals and plants, all of the organisms all of the organisms are composed of the cells. Meaning the basic unit life in the world is cells. Kalau tak ada cells, maksudnya tak ada animal, tak ada plant, semua tak ada hidupan punya, no living organism one. So based on the cell theory, all living things are made from cells. Yeah. All living things include microorganisms, virus, fungi, bacteria, even human, plant, dog, cat, fox, elephant, all these living organisms are made from cells. So cells are the basic unit of the life. Yeah, it is the basic unit of the life, and then new cells are produced from the earlier cells through the cell division. So how the cell produced? It is through cell division, mitosis and meiosis process, right? Even STPM not learn, STPM syllabus not learn meiosis and mitosis, but you already learn in SPM by what is mitosis, what is meiosis, you should know, and cell division cycle, S phase, growth phase, all these you should know lah. Although STPM not learn, but you should know lah. Okay. Then the number 5 is each cells carry habitable information that can be passed through to the daughter cells during cell division. This one is about inheritance theory lah. So all the cells actually contain the habitable information that is our genetic information lah, gene information that can be passed to the daughter cells. So that is why you see that you are look similar from your father and mother. Maybe your eyes is same as mother, your nose is same as father, like that lah. So because of your heritable information, inheritance, genetic information, because your genetic information is come from your parents, either it's come from parents. So you should have the identity or the characteristic that has the same characteristic similar to your father and mother lah, but not all lah, not all same lah, just like got some part is same, got some certain parts of your body are same as your families. And the 6th one is chemical reaction that create life occur within the cells. And then the last one is they are all of the same basic chemical composition. So this one is about chemistry lah. Because in our cells, actually, there are many chemical reaction, metabolism, right? Right. If you already learned chapter 5, chapter 6, you should know metabolism process, aerobic respiration, anaerobic respiration, and also photosynthesis. All of the chemical reaction actually can occur within our cell. So although our cell is very small, but it is very complex. The metabolism reaction is very complex, and also got many things got many things we can learn from the cell actually. So although it is very small, but our scientists, as a scientist, actually we can learn a lot of interesting information from the cells. Okay, so next is about differences between prokaryotic and eukaryotic. Okay, so actually, if you see this diagram, prokaryotic and eukaryotic cells. So this one is, I think, is plant cells. I use plant cell as example, and also bacterial cells. This one is bacterial cells. So if you firstly look, what are the main difference between them? The first difference is flagella, right? They are this one look like tail, right? They are look like the tail, but for the eukaryotic cell does not have the tail. So the prokaryotic cell, it has flagella for the movement. It has this for the support for the movement, and also it has the what we call as what finger. Ini yang itu ini semua ni dia macam look like bamboo, like the hair one. So this one is not the hair. It's not the hair. Actually, we call as finger. So this one is the main difference between prokaryotic and eukaryotic. So and another one is capsule. Prokaryotic cell contain capsule. Eukaryotic does not have one. No capsule one. Capsule. So what is capsule? Later I will explain it. What is capsule? And then if you see the similarity, so what are the similarity between these two cells? They are both have cytoplasm. Both cells have the cytoplasm. Both cells have ribosome. Both cells also have plasma membrane. And then most cells also contain DNA, contain nucleoid that containing DNA one. So these are the four main components that have on the both cells. So which component does not have so prokaryotic is more simple compared to eukaryotic, because prokaryotic the organelles are not much as eukaryotic have. So eukaryotic have nuclear, RER, endoplasmic reticulum, Golgi apparatus, and also mitochondria, like that, all these. But the prokaryotic cell does not have at all. It does not have nucleus. It does not have endoplasmic reticulum. It does not have the Golgi apparatus. Even mitochondria also does not have. Chloroplast, it looks cells. Some prokaryotic, some bacterial cells have the nucleus, have the chloroplast. Some does not have. It depends. But mostly they do not have chloroplast. But also they also don't have chloroplast. But need to depend the organism. So in examination, you cannot say that prokaryotic cell has chloroplast, does not have chloroplast. Eukaryotic cell has chloroplast. This is not too accurate. Okay. But if you want to write about the endoplasmic reticulum, Golgi apparatus, and also mitochondria, yeah, you can write, because totally does not have. So actually, if you see that the DNA material, the genetic material in prokaryotic cell, it does not have the nucleus one. It is net DNA, what we call as net DNA. Dia terus dekat cytoplasm je. Dia tak ada macam eukaryotik, ada satu bulatan untuk cover dia. It have one circle to cover the DNA. But for prokaryotic cell does not have the cover DNA one. Okay, so these are the general differences. So if you see here, also the prokaryotic cell does not have vacuole also. But for vacuole only is special for plant cell, but animal cell does not have vacuole. For plant cell only. But animal cell got some small vacuole for the like starch granule vacuole, the small vacuole only, not like plant cell as big as like this one. Okay, so these are the general differences. So next, we go to the deeper. Okay, so okay, these the differences. So I repeat again, in examination, please don't, if come out differences question, please don't write like this. Wrong. This is wrong format. Don't write like this. This is just the material for you to easier to understand. So please don't write this. Okay. So normally, example organism for prokaryotic is bacteria and cyanobacteria. All of these are prokaryotic. And then eukaryotic are of course plant, animal, and fungi, and some protists are all eukaryotic one. Okay, and cell size. Okay, normally prokaryotic cell is very small compared to eukaryotic. So prokaryotic normally is 0.5 to 10 micrometer only. But for eukaryotic, it is more larger, 10 to 100 micrometer. So it is more larger lah. And for DNA shape, I already mentioned, normally in prokaryotic cell is circular shape. And eukaryotic cell is linear shape. Dia panjang punya. Panjang punya. Yang prokaryotic dia bulatan punya. Dia akan dalam bulatan. And nucleus is, prokaryotic cell does not have nucleus. No true nucleus. It is net DNA. And eukaryotic, it has nucleus bounded by double membrane nuclear envelope. Okay, and then association of DNA, I already mentioned, DNA is net. And then for eukaryotic, the DNA is associated with histone protein from chromosome. So that is why that eukaryotic cell contain chromosome is containing DNA, because our DNA is associated with the histone protein to form chromosome. Why eukaryotic cell want become like that? Because actually our DNA length is very long. If the DNA is not associated to form chromosome, meaning that the DNA length, I don't know, I heard that if our DNA length is linear at all, our DNA length can be around Earth about 3 to 4 times. 3 to 4 times. The diameter of the DNA length equal to the diameter of the Earth. 3 times of diameter of the Earth. So it is very long. So if not, our cell is not capable. You will see that our cell just 100 micrometer, and then you have to, the DNA material need to, all of the DNA material just at the nucleus only. So it's very bad. Okay, and then present or plasmid, this one plasmid is the spatial characteristic also occur in the prokaryotic cell. So actually, what is plasmid? Plasmid is the circular DNA. It also DNA, but this DNA does not contain the genetic information one. It does not have the inheritance purpose one. So this DNA actually does not give function to the prokaryotic actually, but for now state, actually this plasmid actually is very useful in genetic engineering. So for actually genetic engineering, although you are SEM1, but I think SPM you also learn right what is genetic engineering. So maybe you can recap again what is genetic engineering. It is one of the most famous gene technology in the world nowadays to produce the recombinant organism, especially recombinant bacteria. So for SEM3 senior, although I think the school teacher does not teach yet, does not teach the gene technology chapter yet, but you should know lah, because you already learned in SPM. So this one is the spatial characteristic occur in prokaryotic, but eukaryotic does not have this plasmid, does not have plasmid one. Okay, for ribosome, both have ribosome, but the ribosome types are different. So for prokaryotic, it is 70S ribosome. For eukaryotic, it is 80S ribosome. Okay, so these are the main differences. Although they have ribosome, but the types of the ribosome are different. Okay, so endoplasmic reticulum, of course, prokaryotic does not have. Eukaryotic have two types, small and large. And then organelles bounded by double membrane. Prokaryotic also does not have this type of organelles. Eukaryotic have nucleus, mitochondria, and chloroplast are the organelles that have bounded by double membrane layer. So actually, this one I will explain the second topic. Okay, component of cell wall. So this one is also interesting thing. So normally component of cell wall for prokaryotic is made up of myelin, or in scientific term we call as peptidoglycan. This one is one type of the I cannot one type of the basic component for bacterial cell wall. This is the spatial component for prokaryotic cell. For eukaryotic cell, plant cell, it is made up of polysaccharide. So cellulose, one of the example, plant cell is cellulose. For fungi, it is chitin. So cellulose and chitin are the polysaccharide, right? So I write polysaccharide. So actually, in examination, I am not recommend you write cellulose. The plant cell wall is made up for eukaryotic cell is made up of cellulose. Actually, it's not accurate because fungi actually is made up of chitin. Fungi is made up of chitin, not cellulose. So if you want to write this, you should write both cells, plant cells and fungi. So more accurate. Okay. Respiratory membrane. Okay, respiratory membrane for eukaryotic cell, as all of know, it is mitochondria. Mitochondria act as the site for the aerobic respiration. This one you learn in chapter 5. What are the mechanism of aerobic respiration? But for prokaryotic cell, it does not have mitochondria. So which part that can play as aerobic respiration? Prokaryotic cell also have aerobic respiration. Not meaning that it does not have mitochondria, then no aerobic respiration. No, actually it has aerobic respiration also, but the site is mesosome, not mitochondria. It is another structure called mesosome as the respiratory surface. And then chloroplast. Okay, so prokaryotic cell absent, and eukaryotic cell plant and alga have. Cell division for prokaryotic cell is binary fission, and for binary fission. So binary fission meaning that it is the non-sexual reproduction, meaning that the cells, the daughter cells and the parent cells are look totally same, 100% same. So this is what we call binary fission. So they are just copy and paste je. Senang copy and paste. Copy and then paste je. But for eukaryotic cells, solely it does not will totally same because of mitosis and meiosis process. Okay. Okay, so actually just like that. Okay. Okay, so these are all the differences between prokaryotic and eukaryotic. Okay, so now I just proceed to some spatial characteristic of prokaryotic cell wall lah. Prokaryotic cell. So actually, prokaryotic cell wall is very spatial, very different compared to the eukaryotic one. Because eukaryotic just only one type, but prokaryotic it has either gram-positive bacteria or gram-negative bacteria. So the cell wall actually also the cell wall structure is actually also different. So if you see that I already mentioned that the cell wall of prokaryotic cell is made from peptidoglycan, right? So this one is peptidoglycan. Ini tengah punya. For negative, the thickness is thin. For positive, is more thicker. Dia lebih thicker, more thick. Okay, so actually you don't need to differentiate between positive and negative. This one actually, if you want to pursue study in microbiology lah, for your degree, but actually you don't need to know. I just want to show you the structure of peptidoglycan and the basic structure of the cell wall for prokaryotic cell lah. So if you see that it is totally different from the eukaryotic cell, it is more complex compared to eukaryotic cell. Okay. And then another one is capsule. Okay, so capsule is the outermost layer for the bacteria, for the prokaryotic cell. So prokaryotic cell actually got many layers compared to eukaryotic. It contains capsule, and then got cell wall, and then got plasma membrane. It has 3 layers. So what is the capsule? So It is a jelly-like sticky layer of polysaccharide or protein. So it's made up of polysaccharide and protein, and it is jelly-like. Dia macam jelly punya. So you can see that it is transparent. You will see that it is transparent. Right? So normally, if you look under microscope, normally you cannot see clearly the capsule structure, right? Because it is transparent. Either you do the staining. Also, the function is protect against dehydration attacked by host immune system. So normally this one is protect the cell. So actually, if you know that prokaryotic cell normally, if it is pathogenic, when they enter our body, actually it is can cause the disease, right? Some bacteria, it is pathogenic, it will cause disease. So our immune system will fight them. But because of the bacteria got capsule, so our body immune system actually is not very easy to fight them. So some if the people with high immune system, high immunity, normally they can fight. But for the people who have low immune system, sorry, they very hard to fight because of this capsule and also the cell wall. The cell wall also play the main important defense system for prokaryotic cell. Okay, okay, okay. So that's all for basic. This is just the spatial characteristic for prokaryotic cell. Okay, so now I will proceed to the plant cell and animal cell. Okay, I think this one I don't need to wasting many times, much time because I know everyone should know. So I think I don't need to waste many times. So actually, this one is animal cell, this one is plant cell. So what are the main difference? Cell wall lah. Plant cell has cell wall, animal cell does not have cell wall. And then plant cell, the shape normally is fixed and regular. It is rectangular shape, right? Because of the cell wall. But animal cell is irregular shape because no cell wall. And then coloplast, plant cell have coloplast. Animal cell does not have coloplast. Vacuole, okay. Plant cell have large central vacuole, but for animal, mostly absent. Although it is present, it normally is very smaller in size lah. Normally it just act as the nutrient starch granule, starch vacuum like that only. And then food storage normally is for plant is starch, and then animal is glycogen. And central, this one is spatial characteristic for animal. Plant does not have central one. And then plasmodesmata and pit. This one is the cell wall characteristic. If because of plant have cell wall, so it should have the plasmodesmata and the pit. So animal does not have because does not have cell wall. And lysosome, plant cell actually does not have lysosome, but animal cell, it has lysosome. And flagella, so some animal got flagella one. Some animal, not all, some. Some animal got flagella, but not all. And then plant cell totally does not have flagella. So this one depends what kind of animal cell. Okay, what kind of animal cell? So for example, the most famous one, sperm cells. Sperm cell got flagella, right? It has the tail, right? It is one type of flagella. Flagella to make the sperm to swim. Okay, so that's all for plant cell and animal cell. So I think this one is very basic. You should know already. Okay, next is about microscope. So actually this topic, I think 99% of you not focus at all. Maybe. Because at that time, I even pun tak fokus. Ini saya fokus masa saya dekat universiti, baru saya fokus. Dekat sini, STPM saya tak fokus. So, you want focus or not focus, up to you. For my advice is focus, but if you don't want to score, you can skip for it. But as syllabus, I need to teach also. So, microscope, actually, as you know that, microscope, they have two types, light microscope or an electron microscope. So normally your school lab is most of 100% is using light microscope. Light microscope only, because electron microscope is very, very, very expensive. Even in university, you are also does not have the opportunity to use it. Normally, this one you need to further study until master or PhD. Baru awak ada peluang untuk conduct it because it is very expensive and it is very difficult to conduct to operate compared to light microscope. So if you want to learn microscope, actually there are two terms you have to know lah. So these two terms, I think these two terms is the physics why. I think although you are not taking physics in STPM, but I think you have take physics in SPM by. So in SPM, I think got one chapter is about the Miller one. Miller light, I already forget the chapter topic. Reflection and refraction. That topic, I think you still remember. Recap again. Okay. So these two terms are very important to conduct make the microscope become microscope. You know these two actually no microscope at all. So this two terms actually is more physics. So normally if you take physics, I don't know, you guys got maybe you are taking 5 subjects. So if you got taking physics, you should know lah what are these two terms means. So resolution is the ability of the lens to distinguish fine detail and structure. So in simple words, resolution is the satu benda, bukan satu benda lah. As one ability is the noun ability of the lens. So microscope actually is made up of many lens one. So lens to distinguish the fine detail and structure. So meaning that if the microscope have the high resolution, meaning that microscope is able to differentiate the very fine details. So low resolution meaning this microscope cannot go through the very small microbe one like bacteria. So normally your light microscope is not able to see the bacteria one. Only can see the eukaryotic cell normally. Bacteria, virus, and all is cannot one. Cannot see one. So meaning that light microscope has low resolution compared to electron microscope. And then another one is refractive index. Refractive index is the light bending ability of a medium. So it is the refraction. So if you learn in refraction, you will learn about refractive index, right? It is about angle, right? If I'm not mistaken, it is more about the angle, how the light pass through the lens, and then it will reflect how many angles. So this one, you don't need to learn detail. You just want to know the definition only lah. Okay. So now I will proceed to light microscope principle. So why you want to learn these two terms? Because you need to know about the principle. The principle of microscope based on that two terms. Okay, so if you see that this one is the principle of the compound light microscope. So this one microscope that you use in your school lab. Okay, so if you see that it will involve two sets of lens, objective lens and eyepiece lens. So if you see here, this one is your eyes. This one is your eyes. It will mainly contain two lens. So eyepiece lens actually is this ocular lens. So some books mention ocular lens. So actually same. Eyepiece and ocular lens is same. And then another one is objective lens. But you see that there are another lens is condenser lens. So this lens can be appear or not appear. And appear. Look for see the types of light microscope you use. But ocular lens and eyepiece lens and objective lens must occur present in the light microscope one. Okay. So what are the purpose of objective lens? So objective lens is the responsible for both magnification and resolution of the object. So it is the main lens to give the magnification effect. And then eyepiece lens normally is used to produce real image and magnify but not resolve the image. So normally just to adjust that, no, although you have objective lens, but if you see the your microbe, your sample, you will see that normally it's not clear, unclear. So you need to adjust, right? I think you have to adjust. I think your experiment, one of the experiments have want you to use the light microscope in STP assignment. So you should know how to use the microscope. If you see the structure is unclear, you have to adjust, right? You have to adjust your microscope right at the bottom part. So this is the function of eyepiece lens. It need to produce the real image and magnify it. Also, it has the magnification, but it does not resolve the image. Dia bukan resolve the image. Okay, so ocular lens normally in light microscope is 10 times, right? 10 times, right? 10 ganda. And then objective lens dia ada tiga objective. You have three objective lens, right? Got 10 times, 400 times, and then the last one is 100 times, right? I think you should know. Okay, and then the total magnification, yeah, this one total magnification is objective lens times eyepiece lens. So if this one 10 times times 10 times 1000, and then if 10 times times 440, 400, in 10 times times 10, 100, 1000. Okay, so this is a total magnification. And this one, I think your experiment report want you to count, right? Okay, okay. Okay. However, the resolving power of light microscope is limited by wavelength of the light. So why we call it is light microscope, because of the main source. We can see the image is based on the light. Lampu kan, awak buka mikroskop ada lampu kan, dekat specimen tu ada lampu buka kan. Because of this light. If no this light, although you have this, then you also cannot see the image because of this light. So because of the light microscope, the wavelength of the light is normally is limited compared to electron microscope. So normally the wavelength is about 400 to 700 nanometer only. So normally the organism between this that can absorb this wavelength only can see. If not, they cannot see. So hence it cannot resolve the detailed field finer than 0.2 micrometer and resulting in low magnification. So the microbe that less than 0.2 micrometer like virus, and then the organelle, some organelles like ribosome and ribosome, you cannot see through this microscope one. So you cannot see virus one, and also even bacteria, some very small bacteria you also cannot see. So this is why that the light microscope have the low magnification and also low resolution. Okay, the light may bend in air so much that it misses the small high magnification lens. Immersion oil is used to keep the light from bending. Okay, so if you see this structure, so this is the reflection structure of the light. Okay, so this is a comparison between if you not use immersion oil and you use immersion oil and not use immersion oil. So if no immersion oil, the light pass through this gas light, it will be flat, very the bend angle is very high. The bend angle is very high one. Dia akan pergi ke tempat lain. Itu light energy akan pergi ke tempat lain. Dia akan pergi ke sini. Sepatutnya dia akan pergi ke sini. But dia akan pergi ke sini. So that is why we need to letak immersion oil to ensure that the light can pass through this upper part, pass through this lens, pass through the objective lens, through the to the eyepiece lens. Okay, so these are the principle of light microscope. So actually, if you want to learn about light microscope, the most important thing is these two lenses. These two lenses are the main component to the microscope. Okay, Okay. Okay. So I hope no question for light microscope. Okay. So there are many types of light microscope at all. So one of the most example is light microscope lah. Light microscope that you use in the school lab. But also we also have the dark field dark field microscope. Yang ni background dia hitam. You will see this image. This one is the image that you see normally. You see normally you will use your school microscope, you will see this image one. But if you use this dark field punya, awak akan lahirkan image yang black background one. So why got black background? Because it has the opaque disc at here. Dia ada letak opaque disc at here. So this one is black color. It provide the background black background image. Okay. So that's why because of the opaque disc, so only light reflected by the specimen is captured by the objective lens. So bukan semua light, not all the light will be reflected to the objective lens. Only the light reflected by the specimen, only the sample only. Hanya sampel sahaja yang light sentuh sampel sahaja akan pergi ke objective lens. Kalau tak yang pergi ke yang light sentuh yang background ni, dia tak akan reflect punya. Dia akan reflect keluar. So that is why you can see the background image. Okay, another one is about phase contrast microscope, differential interference contrast microscope. So actually, you don't need to know. The principle. Actually, the mostly principle are same. The structure almost the same. They just modify some structure only. They just modify here and me. But normally the main arrangement. They also have the objective lens. They also have the eyepiece lens. They also have the condenser lens. All of the component also have. Just that they maybe got change some part lah. This part they change, this part they change like that. So okay, also we also have the fluorescence microscopy. Dia look macam ni punya. So pretty sangat cantik lah. So actually if you see Campbell, if you got see Campbell, some of the image look like this right. Some of your Campbell buku Campbell kan dia keluar image macam ni, sangat cantik. So ini adalah menggunakan yang ni punya fluorescence microscope. Awak akan lahirkan image like this. Sangat colorful punya. Okay, another one is confocal microscope. So dia look like macam ni. Okay, so that's all for all type of light microscope. Actually got many, but I just list out the nowadays most famous used one lah. Okay, but actually you don't need to know all of these. You just want to know the basic principle of light microscope only. The basic principle. Okay, now we proceed to electron microscope. So you see this diagram. This one is a structure of the electron microscope. So you can see it is very big compared to light microscope. It is very big. So it is more difficult to conduct compared to light microscope. Okay, so electron microscope. So compared to light microscope is using the light, So as the sources, but electron, what we already call electron, meaning it use electron. Dia tak menggunakan light punya. It does not. Dia tak guna cahaya punya. Dia guna electron. So it use a beam of electron focused by electromagnet in a vacuum. Dia guna electron punya. And then the lens used is electromagnetic lens to focus electron beams. So you just imagine that light microscope use light. Dia gunakan light pass through the lens and then go to the eyepiece lens, that we can see. But in electron microscope, it use electron beam. Dia menggunakan electron beam, and also the lens also using the electromagnetic lens. So it cannot use the glass lens already. Dia tak menggunakan kaca punya. Dia guna glass. Okay. Okay, so the electron beam have the short wavelength produce the high resolution at 0.5 nanometer of specimen. So compared to light microscope have the high wavelength, 400 to 700, right? So it is very limited because of this wavelength. So it is very limited. It cannot produce. It cannot help help us to magnify the organism which is very small one. But electron can. Electron because of it has the short wavelength. Because electron beam can produce short wavelength. So it will have the high resolution effect to the specimen. So it can add up to 0.5 nanometer. So this will cause very high magnification of image is produced. So compared to light microscope, it can produce up to 1,000 times. Not 1,000, 10,000, even 100,000 times. So this is the comparison between one of the main comparison. But however, electron microscope only can produce black and white image only. Yeah. For light microscope, you see here, wah, cantik kan? Semua cantik lah, cantik gila lah macam ni. So, light microscope, you can produce the colorful image one. So these not the computing lah, not computing. Really the image produced using light microscope one, original one. It does not have the computer to modify, does not have using PS, does not have not directly not using the Photoshop, Adobe, no, does not using one original image one. But electron microscope, sorry, it cannot produce the colorful image. It can only produce black and white image only because of it is using electron. Okay. Okay, so there are two types of electron you will study: transmission electron microscope and also scanning electron microscope. So both of them are using electron but got some differences lah. Okay, what are the differences? If you see here, transmission electron microscope, it is the electromagnetic condensation lens. For it contain electromagnetic condensation lens you see here. Electromagnetic condensation lens, electromagnetic objective lens, and also electromagnetic projector lens. So semua lens ni dia guna electromagnetic one, bukan glass punya. Not glass. So it have the electromagnetic condensation lens. So this lens is focus the electron beam on a small area of the specimen and illustrate it similar to the function of the condensation lens of light microscope. And then the specimen is placed on a copper mesh grid. So if you see that the specimen, our specimen is sample lah. Our cell sample is letak at here. Placed at here. Electron beam will pass through the specimen and then through the electromagnetic objective lens. Okay. Finally, the electrons are focused by the electromagnetic projector lens onto a fluorescent screen or photographic plate. Okay, so the final image will call transmission electron microscopy. So image appear as light and dark area depending on the number of electron. So it also only can produce the black and white image. And then the magnification power, you will see that you can up to 10,000 to 100,000 times. So compared to light microscope, it's very, very high one. Okay, so this is about the transmission electron microscope. If you see the scanning microscope, so the main difference is at here. Dia ada dekat sini. So it contains secondary electron. Dia akan buat reflection. So electron gun, so similar, electron gun will finally focus electron beam. Electron pass through the electromagnetic lens onto the surface of the specimen, and then it will have the secondary electron beam produced when the primary electron beam knock the electron out of the surface of the specimen. Dia akan Oh, dia akan sentuh spesimen ni dan akan menghasilkan secondary electron. Yang ni dia akan straightforward ni. Dia macam light microscope, just that it change from the electron, tukar kepada electron ni. Dia continue directly kepada image ni, specimen ni, pass through the specimen and then directly to the fluorescence screen. Ini macam screen lah. Awak boleh imagine transmission electron microscope dia macam cinema. Yang ni adalah specimen sampel kami. Yang ni adalah cinema punya screen, big screen punya. Yang ni adalah screen projector. Screen projector. Yang ni adalah big screen punya. Big screen that we will see from this viewing eyepiece. So kami akan tengok yang ni punya. Apa image yang berlaku kami tengok dia sudah appear dekat sini. But for this one scanning, so it calls scanning. So the electron bit will sentuh here, dia akan pass through the lens, so secondary will produce when knock the primary electron knock the specimen, and then it will separate to form the secondary electron, and then the secondary electron are transmitted to an electron collector. So dia akan ada satu tempat untuk simpan untuk menyimpan semua elektron ini, semua secondary elektron ini, and then the image is produced on the viewing screen or photographic plate. So the image called is scanning electron microscope. So the magnification power for this microscope actually is more lower compared to the transmission. It only can up to 1,000 to 10,000 times only. But for the transmission, it can up to 100,000 times. So this is a main difference. Why is like that? Because it already have some image disappear already because of this. Because it is based on the electron collector. So not all the secondary electron will pass through the electron collector one. Some maybe will go to the somewhere else. Somewhere else already. So not all electrons. So that is why the magnification power is not high as the transmission. Okay, so actually these are the image that you will produce under these two type of microscope. So the left hand side is about SEM scanning one, the right hand side is transmission one. So the main difference is scanning one can produce 3D image. It can produce 3D image one. So this is the pollen grain structure under these two microscope. But for the transmission, no 3D image, it only can form 2D image, same as the light microscope. Yang ni light microscope kan, awak nampak macam ni kan. But yang ni, saya rasa awak tak pernah nampak pun. Yang ni adalah awak Google virus image, right? Wah, sangat cantik, right? Like you Google COVID-19 coronavirus punya image, wah, semua sangat cantik, right? Colorful punya. Yang colorful punya semua tu akan menggunakan komputer dia. Dia akan menggunakan satu software untuk color mereka. So actually the original image is like this. So if you put color, it very beautiful. It will appear very beautiful image. So if you Google the virus structure, so if you see the 3D image, normally is using this microscope. They are bio image, but they are using the scanning electron microscope. So this one normally is just for you want to know the basic structure of the virus, but you cannot see the component within the cells. You can see, but cannot see very detailed. So if you want to see very detailed one, you need to using transmission electron microscope. So these are the two main differences between these two microscopes. So I hope you can understand. Okay, now is the differences between light and electron microscope. Okay, so the sources of light microscope is based on the light wave. Sources is from light. Electron microscope is from electron beam. Okay, and then the wavelength for light microscope is very high, 400 to 700, but for electron, it can very small, very slow, only 0.005 nanometer only. And then the lens used or light microscope is glass, but electron microscope is using electromagnetic. And then the specimen, light microscope, you can use the living specimen. Awak boleh guna cells yang survive punya, hidup punya. Macam animal cell, plant cell, if you use the survive punya, awak boleh nampak kan? If you using light microscope, you can see the cell can be walk around the specimen. You can see the cell can walk around. But for electron microscope, you only can use the dead specimen, dead cell only. Hanya boleh guna sel yang mati punya. Dia tak boleh guna survive punya. Although awak pun guna survive, dia akan mati juga. Kerana elektron ini dia akan menyebabkan sel mati juga. So you need to use that cell. So image, light microscope you can produce color image, but electron microscope you only can produce black and white. And then the preparation, light microscope is very easy, it's simple and very easy. But the electron microscope, is very lengthy and complex. You need to prepare the specimen. Light microscope just 1 hour you can prepare it. But electron microscope it requires few hours. Normally require 4 to 5 hours. Just prepare the specimen only. It very take times very complex And then with resolution level, resolution level for light microscope is very low. For electron is very high. Okay, magnification level, normally light microscope is 1,500 times, and electron microscope it can up to 250,000, even up to 100,000. Okay. Cost, light microscope it is very cheap. You want to operate it, you just buy the microscope and then you just open the light. That's all. You just want to use small electricity only. But in electron microscope, when you want to produce electron beam, it is very expensive. Not like light microscope, you just switch on, switch off like that. No. If you electron microscope, if you switch on, the money is burned already. So if you want to use electron microscope, you need to think carefully first. You need to make sure that is success lah. If fail, then you need to spend another money to produce the electron beam again. And then the size, of course, the size of the microscope, normally light microscope is smaller and portable. You can carry it anywhere. But electron microscope cannot. Electron microscope only can at the certain place only. It can only stay at laboratory only. Dia tak boleh bawa angkat sesuka hati, angkat sini, angkat sana, tak boleh. But light microscope awak boleh angkat sesuka hati. Nak pergi mana, awak nak pergi jungle ke, boleh je. Tapi elektron kena. Dia hanya boleh letak dalam satu bilik je. Okay, so that's all for all the differences between light microscope and electron microscope. So I think got question? No question. Okay, so that's all about the first topic, prokaryotic cell and eukaryotic cells. So no question? Okay, so no question. Okay, so if you have any question, you just type on the chat box. See here, no question at all. Okay, so everyone is very expert. So no question. Okay, fine. Okay, so that's all for microscope. So next, I will proceed to the cellular components. Okay, this one, very. I think everyone know about it, right? Organelles. This topic you will learn all about organelles. Semua tentang organelles. Right. And then organelles, maybe many people also feel very bored. I don't know. But I cannot full screen. Wait, full screen. Can anyone hear me now? If no, if yes, can type yes. Hello, can anyone hear me? Okay. Yes. Okay. Thank you. Okay. Sorry because of line. Okay. So now I proceed to the second topic. So this one is actually also the most boring topic. But although boring, you need to know learn also. Okay. What cannot? What cannot share screen? Wait a minute. I don't know why like that. Maybe you guys can have a rest first. I need to settle first this and rest for 5 minutes. At 9:05 we proceed. I need to settle this first. Aiyo, why like that? 9:05 we proceed. Okay, sorry. Okay, now have a rest first. Okay, so now we proceed. Okay, so this chapter cellular component. So this one all we will talk about all of the organelles that we will found in animal cell and plant cells. Okay, so these are all the organelles that you should learn in syllabus. So there are total 4, 6, 7, 10 organelles you have to learn. Oh, banyak kan? But actually all of this you already learn in SPM: nucleus, mitochondria, cytoskeleton, chloroplast, endoplasmic reticulum, lysosome, cell wall, vacuole, Golgi apparatus, and ribosome. So I will explain one by one all of these. So what you want to learn is you need to learn about two things. The first thing is about the structure. What are the structure? What are their structure? And then the second thing is their function. Just two things. So kurang je, tak banyak kan? Mana banyak? Two things je. You need to learn structure and also function. Okay, now first one, nucleus. So this is you can say that it is the heart of the cells, main heart of the cells one. Okay, so nucleus structure is look like this. So this one I extract from Campbell book. So you will have Campbell book can refer. So dia look macam ni. So normally it is the largest size organelles in the cells. Paling besar punya. So it normally diameter is about 5 micrometer. So it is very large. And then it is all in spherical or oval shape. So normally dia look like spherical or oval punya, macam telur punya. And then it consists of nuclear envelope with its double membrane across the nucleus. Yeah. So, sorry. It is also one type of the organelle that have the double membrane. Dia ada double membrane layer. So if you see here, I will carefully here, see here, yang ni, nuclear envelope. So you can see here, actually it contains double membrane. Once inner membrane and also outer membrane. And it has the intermembrane space at here. Dia ada membrane punya. Dekat sel sana, nucleus sana ada membrane punya. And then you will see that it also have the pore complex at here. Pore complex yang itu awak tengok macam bunga punya, macam bunga-bunga punya yang merah punya. Semua is we call as nuclear pore. So actually nuclear pore ni dia ada function dia. Function dia. So what are the function? Later I will proceed. Okay, so membrane is made up of lipid bilayer separated by space of the 20 to 40 nanometer. So it is made up of phospholipid bilayer lah, same as the plasma membrane. So normally the structure is same lama je lah, membrane layer, phospholipid bilayer. Okay, and then envelope is perforated by the pore structure with diameter 100 nanometer, and then the nuclear pore is called pore complex. Okay. Next, okay. This is the structure. Got some 11 things. 11 you can elaborate. And then the nuclear side of the envelope is lined by nuclear lamina. So nuclear lamina is the protein filament. So if you see here structure, let me see nuclear lamina at there. Nuclear lamina mana? Yang ni. Dia dekat sini. But actually you see here, yang ni dia tak ada. Yang ni dia tak ada mention. So nuclear lamina dia like this. Dia macam net structure lah here. Yang ni sepatutnya dia dekat sini. Dekat sini. Dia dekat sini. And then nuclear lamina, what is it? The nuclear side level of envelope is lined by nuclear lamina. So if you see the envelope is the intermembrane space of the nucleus. So at here, so they are line at here. Close and then line at here. So if you using the transmission electron microscope, you will see like this one. Ini semua macam dia look like this one. the net lamellar line the inner surface of the nuclear envelope. So it is the light circular spot. The light circular spot are the nuclear pore. So you see that the nuclear pore is what tengok yang ni light nuclear spore, yang ni semua ni adalah nuclear envelope, nuclear pore. And then all of these yang ni semua ni adalah nuclear lamina. So it is one type of protein filament one. Protein filament. It also have the function, also have the function also. So what function later you will see. For now, we just know the nuclear structure only. Hanya tahu structure dia sahaja. So as you see, as mentioned here, it have the nuclear envelope, nuclear membrane, and then it have the pore complex, nuclear pore, and then within the nuclear envelope, we have the nuclear lamina. Dalam nuclear envelope is membrane, right? Dalam nuclear envelope, dalam itu intermembrane space tu, dia ada nuclear lamina. So it is a lead-like lamina lie in the inner surface of the nuclear envelope. So it lie at the inner surface, inner surface at here, dekat sini. So if you see here this structure saya dekat sini. Ini nuclear envelope, right? Ini nuclear envelope. Ini nuclear envelope. Envelope dia dekat sini. So okay. So nuclear matrix, another one is nuclear matrix is the framework of protein fiber extending throughout the nuclear interior. So if you see here, nuclear matrix actually is around the nucleus lah. Semua nucleus ni yang ini we not call as cytoplasm, kami bukan panggil cytoplasm, we call as nucleus matrix. So nuclear matrix is the framework of protein fiber extending throughout the nuclear interior. So dia dekat inside one. So yang inside, kecuali yang chromatin tu, yang bukan chromatin tu, semua ni adalah nuclear matrix. It is made up of nuclear matrix. And then it carry the genetic information in form of chromosome that contain one long associated histone protein. So you will see that this one is histone protein. When it associated with the DNA, then it will form the chromosome. So this is the form chromosome. So it will form like that. You will see this. Yang ni semua needle light line ni semua adalah chromosome. Okay, and then the complex of DNA and protein form chromosome is chromatin. So if you see here, this one is already compact together to form chromatin already. Kalau awak besar lagi, dia akan menjadi kromosom. Kalau nak besar lagi, dia akan jadi DNA dan histoprotein lah. And then consists of non-dividing core nucleolus. So you see that within the nucleus, we also have the one structure called nucleolus. So this one is the structure dekat tengah lagi, at the middle part. So you see the cell already got one big nucleus, and within the nucleus got nucleolus again. So dia banyak layer lah, satu layer macam ni nucleus, dekat tengah-tengah ni pun ada satu nucleolus. So okay, so these are all the structure about the nucleus. So okay, now we proceed to the function of the nucleus. Okay, sorry. Okay, so what are the function of the nucleus is pore complex, there is nuclear pore. All of the nuclear pore is to regulate the movement of protein and mRNA from nucleus. Okay, so if you not already forget, I recap again about the central dogma theory I already mentioned in chapter 1, transcription process. So actually DNA, our nucleus contain DNA, right? So the transcription process will occur in the nucleus and it will produce the mRNA strand. So this mRNA strand later will pass through from nucleus toward the ribosome. So how it come out? It will come out through this pore complex, nuclear pore. akan keluar dekat nuclear pore ni. Yang ini semua ni. Yang ni. Dia akan keluar dekat sini. So this is the one of dia macam pintu lah. Pintu untuk nukleus. Awak senang cerita lah, pintu untuk nukleus. Okay, the next one is nuclear lamina is to provide the mechanical support to maintain the shape of the nucleus. And nuclear lamina and nuclear matrix is help to organize the genetic material. Okay, so nuclear lamina yang ni, semua ni is to provide the structure one. Dia provide the mechanical support. Nucleus pun kena support juga kan. Kalau tak ada support, nucleus akan bergerak ke mana-mana je. Mana-mana je. So, tak boleh. Kami kena support fix dengan dia. So, siapa support dia? Nuclear lamina yang support dia. And then nuclear lamina also have another function is to organize the genetic material. Right? And then for number 4, nucleolus. What are the function of nucleolus? Nucleolus is the place to synthesis RNA. So our ribosomal RNA is produced at here, and then it will come out also pass through the nuclear pore also. So actually nuclear pore not only mRNA, RNA also will pass through this, and then it will combine with the protein to form ribosome. Okay, and then the protein will import to nucleolus to assemble with RNA into the ribosome, and then nucleus is a place to synthesis protein by mRNA that later will be transported from nucleus for translation in ribosome by tRNA. So actually this one I already explained just now. I say that mRNA is produced in the nucleus, right? So this one the main function in the of the nucleus and based on the structure. So you need to know all these. Okay, so these are all about the nucleus. So any question? No question. Okay. Okay, so this is the first organelles. And then the second organelle, you will proceed to endoplasmic reticulum. Okay, so as we know that we have two types of endoplasmic reticulum, are rough and smooth. So normally the structure is the general structure is it is the network of tubule and membrane bound set structure known as cisterna. Ini structure dia we call as cisterna structure. Dia macam tubule kan, dia macam tube kan. You will see that dia macam tube and then it is membrane bound set. So dia macam membrane bound punya, and then got the structure we call as cisterna. And then these channels are continued with outer layer of nuclear envelope. So normally the endoplasmic reticulum is attached to the nuclear envelope one. Dia berkawan dengan nucleus one. And then the endoplasmic reticulum separate the internal compartment of endoplasmic reticulum called cisterna space. So within the space ni, this space we call as cisterna space. Awak tengok kan, yang ni dia ada satu space kan. So this one we call as cisterna space. Okay. Okay, so about the differences between RER and AER in terms of structure. So rough punya, of course, why we call rough because of it contain ribosome. So you see this structure yang ada titik-titik semua titik ni ada ribosome. So ada titik-titik ni semua ni adalah rough endoplasmic reticulum. So because of it have the ribosome attached to them. Okay. So it has the flattened set tubule. And then SER is not flattened set, it is the interconnected tubule and not flattened set. Ini key point yang awak kena alert. It is the interconnected tubule. What does mean by interconnected? Meaning dia macam pipe punya. Senang cerita. Dia macam pipe punya. Compare to rough. Rough bukan pipe. Rough is the flattened set. Dia macam kertas. You imagine lah, dia macam kertas punya. But in SER, dia macam tube punya. Memang macam tube. So what we call as interconnected because they are interconnect together. Mereka akan interconnected together. So dia memang look like tube punya. Okay, so it does not have ribosome attached to it. So for smooth endoplasmic reticulum, it does not have the ribosome. Okay, so that's all for the structure of endoplasmic reticulum. And then we go to the function. Okay, so for rough endoplasmic reticulum, so it is the main function is to synthesize protein from ribosome. Protein synthesized from ribosome will attach to the RER. Okay, why it attached to IR? And then IR. So later, this protein form polypeptide protein will enter the cisternal phase and fold into its functional shape. Maksudnya RER ni dia akan provide the shape of the protein. If you still know that our protein got four structure, right? Primary structure, secondary structure, tertiary structure, and quaternary structure. So the protein synthesis by the ribosome, actually just the single sequence only, polypeptide sequence only. So if you want to give the shape for the for the protein, we need rough endoplasmic reticulum. Rough endoplasmic reticulum will fold the polypeptide to become its functional shape. Okay, next it will form the secretory protein. Glycoprotein is formed through the attachment of the carbohydrate and polypeptide. Okay, and then the secretory protein will trap in the membrane of the vesicle that is bud-like called transitional endoplasmic reticulum. So if you see structure, this is what we call as transitional endoplasmic reticulum. Yang ni. Meaning this one, yang ni, meaning that it already have the functional protein already. It will begin trying to form the vesicle called transport vesicle already. So this transport vesicle will contain that protein, that glycoprotein. So in general, senang je, tak perlu risau. I already mentioned nucleus will produce our nucleus will use the DNA material. Inside the DNA material will produce mRNA, right? And then mRNA dia akan come out pergi ke ribosome. Pergi ke ribosome, nanti I will explain. Pergi ke ribosome, ribosome tu akan hasilkan protein. Dia akan hasilkan protein. Okay, after ribosome hasilkan protein, dia akan masuk ke rough endoplasmic reticulum to the cisternal space, to make the protein become the functional shape. Akan menghasilkan functional shape. Then this protein akan attach to the carbohydrate to form secretory protein, glycoprotein. Okay, then this secretory protein will akan break dengan membrane ni, akan form transitional endoplasmic reticulum. Okay, then this transitional endoplasmic reticulum akan hasilkan bud or to form transport vesicle to transport to the Golgi apparatus for further function. So it is the one of the key organelle in protein modification, not protein synthesis. It is more to protein folding. So I think in SPM, normally in SPM punya buku just bagi tahu awak or RER, synthesize, modified, form protein only. But in STPM, you cannot write the general function like that. You need to explain all like this. These are all the functions that will occur in the rough endoplasmic reticulum. It also, when the exam come out, want you to answer how protein is synthesized. You need to include this answer. Not only include this, you need to include the original one in the nucleus The mRNA is synthesized and then go to ribosome and then go to here and then will go to Golgi apparatus for further modification. You need to write until there. Memang kena tulis sampai sana. So very long one. So I think got 1 year right. Got 1 year. Dia essay question tulis dia keluar soalan macam ni. So dia keluar soalan macam ni. Normally it is very easy to write if you really know all the theory about all the organelles, what happened in that organelles. So these are all the main functions for the RER, rough endoplasmic reticulum. So hopefully you guys can know. Okay, so this one, okay, so next is about SER. So smooth endoplasmic reticulum. So this reticulum is totally different for RER1. The function is totally different. It does not have any relating function for protein. Tak kait dengan protein langsung. So this function is it contains the enzyme which is important in lipid synthesis, like oil, steroid, and phospholipid. So it contains a specific enzyme that can hydrolyze the lipid one. So it also has the metabolism of carbohydrate. Some metabolism of carbohydrate here. And it also contains the enzyme that can detoxify the poison and drug by adding OH group to making them more soluble and easier to flush from body. So it also acts as a detoxification. Kalau ada toksik masuk ke dalam body kita, SER akan play the function to help our cell, our body to detoxify this drug. But not say that awak boleh makan drug lah, makan merokok lah, semua tak boleh, bukan. Just that it has the function like this. Okay, and also it is act as the storage of calcium ion. So our calcium, semua calcium ion akan simpan ke sini. Bukan simpan ke muscle bukan simpan ke bone kita, bukan tulang kami. Although tulang kami ada store calcium, but for the muscle cell actually does not store any calcium one. Actually is SER store the calcium ion. Why muscle want calcium? Because of contraction. Itu filamental hypothesis. Yang itu semester 2, SEM 2 awak akan belajar. Yang senior, you should know lah. Muscle contraction mechanism. Muscle contraction mechanism, right? At chapter, if not mistaken, at chapter 3, right? Chapter 3, semester 2. Chapter 3 I already forget semester 2 lah. Semester 2 you learn. Okay. Storage of calcium ion. So it can pump the calcium ion from cytosol to endoplasmic reticulum lumen, and then when the muscle is stimulated, calcium will rush back to the cytosol, trigger the contraction of muscle cell. So these are the 4 main functions of the smooth endoplasmic reticulum. So the function is totally different from the rough endoplasmic reticulum, totally different. So not be confused. So hopefully you can differentiate them properly. Okay, next organelles is about Golgi apparatus, or another book they mention Golgi body. So both of them are correct, but I recommend you use Golgi apparatus lah. Okay, so Golgi apparatus, about their structure, actually if you see that structure, it looks similar like the endoplasmic reticulum one. Macam sama aja, tapi not 100% same, because it normally same as the rough endoplasmic reticulum punya. But just that rough endoplasmic reticulum ada ribosome, yang ni tak ada. So it consists of a stack of flattened membrane bound set called cisterna. So dia macam sama dengan rough endoplasmic reticulum cell. But it is not interconnected lah. So it is not the smooth one, not interconnected one. And then each cisterna is covered with a layer of lipoprotein membrane. So if you see here, actually it it is covered by the lipoprotein punya. Semua layer tu dia akan each cisterna is covered with a layer of lipoprotein membrane. So normally the layer here is made up of lipoprotein. And then the cistern Golgi apparatus got concave shape and also convex shape. So please differentiate them. And when you draw, you also need to draw the concave shape and also convex shape one. So the concave shape is known as the trans. So yang ni concave. Kan, yang ni concave dia macam sangat lengkok kan. Convex dia bukan lengkok kan, convex dia macam bulat ni, macam ni bulat ni kan. Concave dia macam lengkok sangat dalam kan. Convex dia bukan, dia bulat macam bulat kan. Okay, so this is the main difference. You have main key point in Golgi apparatus structure you need to know. So concave shape is about transphase. As trans Golgi network near the surface of plasma membrane. So dia dekat plasma membrane punya. It will face to the plasma membrane punya. Yang convex punya, yang ni, dia akan dekat near the surface of the endoplasmic reticulum punya. Dia akan face dekat dalam punya. Face dekat nucleus lah, senang cerita. Dia akan face dekat nucleus punya. So if examination want you to draw, you need to draw carefully. You draw the balik, concave, you draw the convex shape dekat dalam, concave shape dekat luar. Maksudnya totally salah lah, because you already wrong theory already wrong. So your drawing is no marks at all. So you need to differentiate between them, or in examination maybe instruction question their point at here. What is this phase? Transphase or called transphase or cisphase. Transphase is convex shape, while cisphase is concave shape. So cisphase is convex shape, transphase is concave shape. Okay, so please differentiate them better. Okay, so these are only the structure that if you answer structure, you need to answer like this. Okay, next is about the function. So what are the function of Golgi apparatus you can write? So actually it is just proceed to the function of the protein synthesis that we come from the endoplasmic reticulum. So endoplasmic reticulum will bud off to form the vesicle, right? Transport vesicle, right? So the transport vesicle, this one, you see here, this one, dia akan move toward the Golgi apparatus. It will move toward the cis phase of the Golgi apparatus. Cis phase not transphase. Kalau awal tulis transphase totally salah lah, tak ada masalah. Kadang awal dah wrong theory. Cis phase, dia akan masuk ke cis phase, and then the vesicle will be collapsed, will bound together to form the new cis Golgi cisterna. So after that, what are the protein inside here? Apa protein tu buat? Kerana itu inside ada protein tu buat apa? Kenapa dia nak masuk ke sini? So this one is just to store and modify the protein in cisterna and then transit from cis phase to trans phase. So yang ni cisterna maturation, Golgi cisterna move in a cis phase to trans phase direction, and then vesicle form the okay. So the protein inside here, dia akan masuk dari sini kan. So protein akan transport from cis phase to trans phase. So apa yang akan buat? Akan store lah. Either protein store at here or modified. Dia akan modify again protein. And then later on, it will produce a secretory vesicle contain hormone and protein and release contained by exocytosis. So if you see here, Actually, this one just RER to their function untuk make the protein ada shape, fold the protein become a shape. This one to modify the protein become the protein that at the specific function. Like we know that we have many type of protein, right? It can be function as both hormone, it can be function as the transport protein, it can function as antibody, it can function as many types again. So this one is to modify. Dia akan modify je. So it maybe can form hormone, maybe can form insulin, glucagon, core protein, transport protein, or even antibody like that, all of that. Okay, and then after that, it will leave the Golgi apparatus, and then transport the protein will be transported in within the secretory vesicle. Tadi is transport vesicle. Sekarang ni adalah we call as secretory vesicle. So yang dalam ni tak ada protein yang sudah really boleh function lah. And then after that, it will transport to the plasma membrane, transport toward the plasma membrane, and release that protein to the outside of the cell. So these are all the procedures from nucleus until Golgi apparatus until outside of the cell. So if exam want to ask you like this, you need to mention all of these processes directly. Okay, so vesicle form and leaf Golgi carry specific product and protein to other location or to the plasma membrane for secretion. Okay. Also, okay. So next is vesicle transport some protein backward to less major where they function. Vesicle also transport certain protein back to the endoplasmic reticulum, their site of function. So actually, not only secretory vesicle not only transport to the plasma membrane, it also may be transport to other part like endoplasmic reticulum, because endoplasmic reticulum is the organelles. It may be also need certain protein also. Maybe it requires certain protein for the function also. Okay. Put some protein backward. And also maybe the vesicle protein also will transport to another Golgi apparatus. Because this is the major of Golgi apparatus. Maybe some cell, just a new cell, the Golgi apparatus is not major. So maybe go to another part, another new Golgi apparatus for their function. So these are the vesicle can be go to three ways. Got three ways. The first way is go to plasma membrane and then secrete out from the cell. And another one is go back to the endoplasmic reticulum for another function, And then another one is go to the new Golgi apparatus, another type of Golgi apparatus, especially the immature of Golgi apparatus to make them major. Okay, so these are all the direction of the vesicle, and then it also can form lysosome. I think this one everyone know. Everyone remember this. Yang lain semua tak remember punya. So actually just like that. Okay. So that's all for all the protein synthesis. If examination want to you mention about the protein synthesis, how protein is synthesized, you need to mention from nucleus until go to the ribosome, go to the endoplasmic reticulum, go to the Golgi apparatus until go to the secretory vesicle and go to the that three direction. So actually got many things to write So actually it come out like this, is a bonus to you I think. Very easy. And okay, another Golgi apparatus function. Fusion of secretory vesicle with plasma membrane to add the surface area of the membrane. And then in plant cells, polysaccharide is secreted for formation of cell plate and cell wall, and also it is the formation of peroxisome. So for plant cell, it's spatial. It can produce the polysaccharide to form the cell plate and cell wall. Okay. So next. Organelles is lysosome. So as I mentioned, lysosome is come from is produced for Golgi apparatus. Okay. So lysosome is actually the spherical structure, small spherical sac structure. It looks like this. And then the special is it contains the hydrolytic enzyme, normally protease and lipase. And then this content of lysozyme are acidic because it will work best in acidic conditions. So normally within the lysosome, the enzyme here normally is very acidic one. And then the hydrolytic enzyme is made from RER and then transported to Golgi apparatus for processing. So this one you can imagine that it is come from the secretory vesicle. But this one is not will form the secretory vesicle, it will not release the enzyme, it will form another organelle called lysosome. So after it modified in the Golgi apparatus, and then it butt off, it will not form the secretory vesicle, but it will form the lysosome. So normally it is formed primary lysosome. Actually, we have primary and secondary lysosome. So primary lysosome are formed by budding from Golgi apparatus. So in specific, actually at function of Golgi apparatus, you should write formation of primary lysosome, not just lysosome. You need to mention primary. Very need to specific lah. Okay. And then these are about the lysosome function. So actually lysosome function, the first function is to digest the food and waste product in form of food vacuole and phagocytic vacuole respectively through endocytosis. Okay. So if you got the food vacuole, kalau ada makan makanan, makanan ni, so dia akan pass through the plasma membrane, enter into the cell in form of food vacuole, and then the lysosome, because it contains the hydrolytic enzyme, it will fuse together with food vacuole and then will cause the digestion. So lysosome will fuse with the vacuole to form the secondary lysosome. So this one we already called known as secondary lysosome. And then the enzyme will release a particle to the food particle and then to hydrolyze that particle. And then so this process is known as phagocytosis. The lysosome akan makan food vacuole tu, so dia akan phagocytosis lah. So this process is known as phagocytosis. Normally this one is occur in protista. Normally is occur in protista. Normally is like Paramecium and Amoeba, the most famous one. Okay. The next function is also lysosome also can makan, also can engulf or digest the unwanted organelles. So the unwanted organelle, whatever, mitochondria, Golgi apparatus, whatever at all, dekat sel. So kalau mitochondria tu dia sudah tua, tak boleh function lah, dia tak boleh function lah. So tak boleh function, lysosome ni dia akan makan, tak boleh function to engulf these organelles, makan mereka. So these organelles are enclosed by a single membrane form autophagosome that later fuse with lysosome to form autophagic vacuole. So this one are formed to single membrane to form autophagosome and then fuse together with lysosome. So this one we already call autophagosome, and then it fuse with lysosome to form the autophagic vacuole. And then lysosome will release the enzyme, and then it will cause the digestion. So it will digest the unwanted organelles. So this process we call as autophagy. So what is autophagy? It will engulf, digest the unwanted lysosome. Ini untuk organelles ini. And another one is called autolysis. Okay, so yang ni dia akan makan whole cells. Yang tadi autophagy tu dia akan makan organelles only. Apa tu organelle dia makan je. But autolysis is the self-digestion of cells. Dia akan digest whole cell. Itu maksudnya dia akan kill cell lah, senang cerita. Matikan sel tu. Normally when the cell is old lah, is already unwanted, already tua, tak boleh function already. Occur when the cells are die. Normally if cells is die. So normally so this is about the autolysis, and also lysosome is also play the role in defense system. So normally if they are the phagocytic vacuole rather than the food vacuole, kalau ada phagocytic bacteria tu yang ni lysosom dekat leukocyte, one type of white blood cell, right? White blood cell tu ada lysosom tu, dia akan pergi ke phagocytic vacuole dan makan dia, engulf dia by releasing the hydrolytic enzyme to rupture the cell wall of bacteria to kill the bacteria. So this one process actually same as this, but just difference is the food vacuole is replaced by phagocyte pathogenic vacuole. So both process are also known as phagocytosis. Just the difference, one is food and another one is pathogenic vacuole, bacteria lah. Okay. So, okay, that's all about the lysosome. Okay, fifth organelles is about mitochondria. Okay, mitochondria, I think the structure is look like this. So actually it is spherical in shape, and normally the length and width is like this. And then it is the another type of double membrane organelles. Nucleus is double membrane, right? Mitochondria is another type, which is double membrane also. So it has the intermembrane space and matrix. So if you see here, dekat sini, see here, intermembrane space lah. Ini adalah intermembrane space. Yang ni merah ni lebih shining ni adalah outer membrane. Dan kalau dekat sini, it is an intermembrane space. So it also have it has the crystal and the matrix. In the matrix, and if you alert that in mitochondria also contain the DNA. So meaning DNA not only occur in the nucleus, but found in the nucleus, but also it can also found in the mitochondria also. So this one you have to alert. Okay. So inner membrane has the folded crystal. So the crystal is folded and the outer membrane is smooth. And matrix is enclosed by inner mitochondrial membrane, consists of DNA, protein, and also ribosome. 70S ribosome. So this ribosome actually same as the prokaryotic cells one. And there are many stalked particles on the surface of crystal. Okay, now about the function. So normally function of mitochondria is about the carbohydrate metabolism, which is aerobic respiration. So actually aerobic respiration you will learn in chapter 5, right? I think your school teacher already proceed, I don't know. Okay, so outer membrane control the movement of substance from mitochondria, and then crystal increase the surface area of inner membrane, providing the chemical for electron transport chain. So you see that the crystal is folded. Why it want to fold it? Because it need to increase the surface area so that they can have more places to provide the electron transport chain. So it can provide to produce more ATP lah. And then the matrix contains the enzyme were involved in the reaction of Krebs cycle. So Krebs cycle is occur at the matrix. And then the outer and inner membrane are permeable to hydrogen ion. So this hydrogen ion is also very important in terms of oxidative phosphorylation and chemohomeostasis, right? Okay. So so this one you should learn in chapter 5. You will learn more deeper in chapter 5. Okay. Next one is ribosome. Okay, ribosome, I already mentioned there are two types of ribosome. So one type is 70S, another type is 80S. So 70S is normally found in prokaryotic cells and also mitochondria and chloroplasts. And then eukaryotic one is ATS. It is found in the eukaryotic one. Okay. Okay, so the structure of the ribosome is look like this. So this one is ATS1. So it is a spheroid shape, not spherical, spheroid. That look like this. And then it found in all cells. So semua cell either prokaryotic or eukaryotic, semua ada ribosome. And then normally it cells that produce a lot of protein such as granular cells. So the cells that produce a lot of protein will have more ribosome because ribosome is a main function to produce protein. And then the subunit can be attached to form bigger functional unit in the presence of magnesium ion made of RNA and protein synthesized in the nucleolus. Okay, next about the function. So normally ribosome function just only one. It provides the site for the formation of peptide bond in which amino acids are joined to form polypeptide. So in general form, it is play a role in protein synthesis. But in specific, you need to write like this. Not say the E play a role in protein synthesis. You write at here in exam normally fail, no marks at all. Too general. But this one actually you will learn in translation process. Semester 3, translation process. So this one actually you will more clear when you learn translation mechanism. But unfortunately you are only sem 1, so you don't need to learn. After you enter sem 3, you will learn. So it will provide the site for the formation. You see here, ribosome dia ada banyak lubang macam ni kan. It is the site to ensure that tRNA boleh bind dekat sini. And then after dia bind dekat sini, it will form the peptide bond with the amino acid. Amino acid. Dia akan form the peptide bond together to form the polypeptide chains. So in general it just to provide the site to form the peptide bond. Just like that. And then to form the polypeptide. Okay, next is about chloroplast. Okay, chloroplast. Okay, chloroplast actually is the spatial organelles that only founded in plant cell. Animal cell does not have chloroplast. So chloroplast is the biconvex circular shape. And then it also have the double membrane, bounded double membrane. So altogether got 3 organelles that have bounded double membrane: nucleus, mitochondria, and also chloroplast. These 3. And then it also contains the DNA, 78 ribosome, and also starch granule. And then it also have lamella is suspended in the stroma. So we see this structure. Maybe not clear. So you see this structure, lamella is here. It's this. This is lamella. Okay. So if you see that lamella is suspended in the stroma. So in chloroplast, you need to differentiate between thylakoid and the stroma, and also granum and stroma. So granum is the yang ini punya, yang ni thylakoid. It is the composed of the thylakoid, and then the stroma is the outer surface of the chloroplast. So yang surface ni semua ni we call stroma. Yang ini thylakoid ni, kalau semua thylakoid composition of thylakoid combine together, we known as granum. So you need to differentiate between them. What is granum and thylakoid? Granum and stroma. And then so lamella consists many flattened disc-like and fluid-like set is thylakoid. So if you see here, normally this one is thylakoid. Thylakoid yang ni one unit. Not all these, one unit ni we call as thylakoid. And then stack of thylakoid will form the grana. And then grana is connected with another by intergrana lamella. So they are you will see that you will see here they are connected together, right? They are satu connected. Grana is connected together by intergrana lamella. And then chlorophyll pigment is found within the thylakoid. So these are all the structure about the chloroplast. Okay, next one is about the function. So normally the function is undergo photosynthesis. Normally produce the organic compound glucose molecule through the photosynthesis process. And then it can store excess starch produced from photosynthesis because it contains starch granule. And then the thylakoid is the site for light-dependent reaction, and then the soma is the site for light-independent reaction. So what are these? You will learn more detail in chapter 6, the last chapter in this semester, photosynthesis. Okay. Next organelles is about vacuole. Okay, this one is also the spatial organelles found in plant cell. So it is the fluid filled set bounded by single membrane called tonoplast. Actually, vacuole is the fluid filled set fluid filled set one. So it contain the fluid lah. Fluid yang dia akan ada banyak liquid lah. Banyak liquid, and then it is bounded by the single membrane called tonoplast. So the membrane of the vacuole we call tonoplast. And then the fluid in vacuole is called cell sac that consists sugar, mineral salt, and waste. And then vacuole found in animal or alga are known as quantical vacuole that is smaller in size. So animal and alga also contain vacuole but very small compared to the cell. Compared to the plant cell is very big, but animal cell is very small one. And then some organisms also consist of food vacuole formed from phagocytosis. Ini yang tadi saya cakap dengan phagocytosis, right? Okay. So what are the function of the vacuole? So the function of the vacuole for the animal cell one, quantical vacuole is to pump the excess water out from cell to maintain the suitable concentration of ion inside the cell. So normally in animal cell, it is just to play the role to maintain the concentration of ion inside the cell. Kalau banyak, dia akan pump water lah, because normally quantical vacuole is more composition is about water, right? So if the solute concentration is very high outside the cell, dia akan pump water out to prevent the denaturation. Okay, carry out enzymatic hydrolysis function as same as lysosome. So it also contain enzyme also. Small vacuole in plant can hold reserve of important organic compound and protect the plant against herbivore by storing compound that are poison. And then contain the blue and red pigment of better help extract pollination from insect. So dia boleh membantu, dia contain the blue and red pigment to extract the insect for pollination. So this is about the reproduction for the plant. And then the central and then it also contains central vacuole play major role in growth of plant cell that will enlarge when absorb water. Okay, so these are all of the function. Okay, next is about the cell wall. So it is also the spatial characteristic in the cell, in the plant cell. Animal cell does not contain cell wall. Okay, now is about the structure of cell wall. No question. Okay. About the cell wall. So if you see here, cell wall is much thicker compared to the plasma membrane. Yeah, of course. Cell wall is memang dia thick lah, sangat tebal punya. And then the cell wall in plant cell is made from bundle of cellulose known as microfiber. So if you already learn, I already teach about the structure of cellulose, right? When the cellulose is combined together, the average antiparallel combined together, they will form the microfiber. And then microfiber is synthesis from the enzyme called cellulose synthase. And then microfiber will be secreted to the extracellular space embedded into the matrix of pectin and hemicellulose. Primary cell wall first secret in young plant cell is thin and flexible. So when you see here, our plant cell wall has primary cell wall and secondary cell wall, right? Cell wall, right? So secondary cell wall is more thicker compared to primary cell wall. You see this structure. You see this structure. Primary cell wall just yang merah ni je. Secondary cell wall more structure. So normally the plant that have secondary cell wall is the woody plant. Normally the non-woody plant only have primary cell wall only. Woody plant, trees, for example, trees that can grow very tall one, is the got secondary cell wall. But like the plant like pea plants, hibiscus, not hibiscus, pea plant, orchid like that, does not have the secondary cell wall. It only have primary cell wall. So hygrophobia will be secreted to the extracellular space embedded into the matrix. Okay, and then the adjacent between the primary cell wall is known as the middle lamella made for pectin. So I think I need more detail. So actually, if you see here, see this structure. This is about the primary cell wall. So if you see here, this one yang kuning ni, yellow is we call as cellulose microfiber. This is made up of cellulose molecule through the cellulose syntax. So after the cellulose, all of the cellulose molecule, they antiparallel to each other, and then they pass through the cellulose syntax. They will form the cellular microfiber like this, block shape, right? And then there are also the hemicellulose. So hemicellulose, it is not cellulose. Dia bukan cellulose, it is the component, many component of carbohydrate one. So it has xylene, mannose, zygoglucan, and then beta glucan. So this one actually it will support the hemicellulose. So that is why it say that microfibrin will be secreted to the extracellular space embedded into the matrix of pectin and hemicellulose. So microfibrin ni akan pass through the extracellular space. Yang ni adalah extracellular space. Ini adalah satu space yang besar untuk komponen untuk sel wall. So it has the plasma membrane, right? Ini plasma membrane, and then ini cell wall. Yang ni pectin. You see that pectin also is the another component. Ini komponen dia lah. You don't need to memorize it. You just the component is pectin. Okay, so the adjacent between the primary cell wall is known as the middle lamella made from the pectin. So you see that if you refer that the adjacent between the plant cell wall, plant cell wall dia akan mungkin dia akan combine together. Macam ni kan, combine together. So kalau combine together, dia ada tengah-tengah line ni we call as middle lamella. So middle lamella is made from the pectin. So dia dihasilkan daripada pectin. So inilah middle lamella. Dan ni satu line that act as the adjacent between to separate the cell wall between the adjacent cell. So this one how it can be produced? It is produced from the pectin. Okay, in the pectin one. Okay, so that and then when the cell go, the cell will strengthen its cell wall. Then secondary cell wall is added between the plasma membrane and primary cell wall. And secondary cell wall has the strong and durable matrix that can give strong protection and support. And cell wall is perforated by channels between the adjacent cell called plasmodesmata. So if you prefer this structure, primary cell wall is more thin compared to the secondary cell wall. So if you see the secondary cell wall, you see that the main difference is the cellular microfiber is more thicker compared to the primary cell wall. So it more thicker and then it contains another component called lignin. Lignin already. So actually primary cell wall is at here already. Dekat sini lah. Dekat sini. Kecil-kecil je. Ni dah merah already mentioned right. Mentioned that secondary cell wall is added between the plasma membrane and primary cell wall. So between the plasma membrane and primary cell wall. So inilah semua secondary cell wall. So the main difference secondary cell wall is it contains more thicker cellulose microfibule, and then it contains another component called lignin. So this lignin actually provide more support, more protection, mechanical support to the plant. Okay, so these are all about the cell wall. So these are the structure and differences between primary cell wall and secondary cell wall. And you will see also primary cell wall, the arrangement for the cellular microfiber is loosely, and for the secondary cell wall is more packed together. Okay, and then cell wall function. Of course, cell wall is to provide the mechanical and skeletal support for the plant cell, and then it can form the secondary cell wall to increase the strength of the cell, become more rigid, and then cell wall is rigid. When cell wall is rigid, it will be resistant to expansion. So this one normally is when the plant cell is involved in the hypotonic condition. So although I not teach chapter 3, but I assume that all of you know what is about hypotonic. So when the plant cell is immersed in the hypotonic condition, meaning the water potential outside the cell is very high, right? So water will move into the cell by osmosis. And then in theory, in your SPM, you learn that plant will not denature, will not burst compared to animal. Animal will burst, right? But plant cannot burst. Why? Because of this cell wall. So because of this rigid cell wall, so it protects the cell against from bursting. So plant cell will not burst although it is fully with water. Okay, and then arrangement of the microfiber also help to control the cell growth and shape. That microfiber also can control the cell growth. So because the ability to shape the cell is determined by its arrangement. So normally, I already mentioned that primary cell wall is randomly arranged to form loose network. So primary cell wall is not loose, it's loose. So normally the growth and shape of the primary cell wall is not very, for the plant, they only have primary cell wall is not very tall because of the limited, because primary cell wall is not straightened compared to secondary cell wall. So this makes the cell wall more elastic and allow stretching during the growth. So actually arrangement of microfiber is the key role to make sure that either the cell wall is elastic or stretching or not. So for primary cell wall is less elastic compared to secondary cell wall because the arrangement is not very good. It's loosely networked. Okay, next one is about the plasmodesmata. Why we have the plasmodesmata? So plasmodesmata is about the transport substance between the adjacent cell. So because of the cell wall, it's not like the animal cell. Animal cell, because only have plasma membrane, although they bind together, they closely together, they also can transport substance easily. But because plant cell have cell wall, it's not easily. So it require plasmodesmata, dia macam pintu juga lah. Pintu untuk cell wall, so that the substance within the cell can be easily transported to another plant cells. And then the cell wall is coated with waxy cuticle to reduce water loss and risk of infection, and control the excessive or loss of water from epidermal cell of leaf and stem cell by having a waxy cuticle layer. And then provide food storage in the form of the hemicellular in some sense. So these are all the cell wall function. Hopefully you can memorize all. So there are total 7 functions. Okay, you should memorize all. Okay. And then last one is about the cytoskeleton finally. Now it's already, oh, we already 10:10. Okay, so sorry because this chapter is very long, very long. So I hope you guys still with me. Let's see, still with me. Okay, okay. So, okay. Now I will proceed to the cytoskeleton. This is already last organelles, but also the longest part. So want to fast also cannot. Okay, so I try to end by 10:30. Okay, cytoskeleton. So this is the structure of the cytoskeleton. So what is cytoskeleton? So cytoskeleton actually is one type of organelle that is a network of fiber extending throughout of cytoplasm. So in the surface, actually you cannot see the cytoskeleton. It actually is inside within the cell one. Awak kena tengok sel kan, ni sel kan cytoplasm, dia dekat sini punya, dekat bawah-bawah punya. It is the extended network fiber throughout the cytoplasm. So dalam cytoplasm semua tu ada cytoskeleton. So if you see this diagram, so this one is the using fluorescence microscope lah. Fluorescence microscope, just now we just learned already. Fluorescence microscope will give this image. Okay, the most function of the cytoskeleton is giving mechanical support to the cell and maintain its shape. So actually cytoskeleton, why we need it? So it may provide mechanical support. So actually cell is also 3D structure. Actually cell is 3D one, not 2D. Because if you see the light microscope is 2D, actually cell can be 3D shape. So 3D shape it need the support, mechanical support. Kalau tak ada support, macam mana sel tu boleh bergerak ke sini ke sana kan. And then the cytoskeleton also provide encourage for many organelles and cytolytic enzymes. So it also provide the organelles to fix the organelles on the cytoplasm. If no organelle, it very easily come out from the cells one. Dia tarik to provide the anchor to the organelles. And then cytoskeleton is more dynamic than animal skeleton because it can dismantle in one part of cell and reassemble in new location, change the cell shape. Okay. This one is about the cytoskeleton structure. Also, if you see here, actually cytoskeleton it contain the motor protein. So cytoskeleton also involve the cell motility, which help of the motor protein. And cytoskeleton and motor protein will work together with plasma membrane to allow the whole cell move along fiber outside the cell. So this will actually to ensure the cell can move. Kan awak tengok sel tu dia akan bergerak kan? Macam mana dia bergerak? Because of this motor protein. It will provide the motor protein adhere and then to provide the cell work together with plasma membrane to allow the whole cell move along the fiber. And then the vesicle and organelles will use the motor protein fit to walk in the cell and their destination along a track provided by cytoskeleton. Also, not only cell can walk, the organelles and the vesicle inside the cells also can walk. So just imagine vesicle. I just mentioned about the transport vesicle and secretory vesicle. How they can move. Transport vesicle move from the vas endoplasmic reticulum to the Golgi apophyseal, right? How it can move because of this. Ini dia pantu, cytoskeleton dia pantu dia. So you see there, ini vesicle. So if you see there, so this one is cytoskeleton, yang bawah ni cytoskeleton, and then dia ada motor protein. It will bind to the vesicle, and then dia akan walk, dia macam walking together, assume ATP lah. So energy dia macam can walk the vesicle along the microtubule or microfilament. Okay, this is just the general characteristic in the cytoskeleton. Actually, there are 3 main types of cytoskeleton you have to learn. Microtubule, microfilament, and also intermediate filament. Okay, what are they? So these three types are also cytoskeleton at all. It also provides support, but there are but the structure are totally different, and then the function is also totally different. Okay, so for microtubule, so microtubule actually is look like this. Dia macam cylindrical shape punya. Cylindrical shape punya, cylinder shape punya. Macam pipe lah. You can imagine it is look like a pipe. Dia macam pipe punya, water pipe. It is the hollow blocks made from the globular protein called tubulin. It is made from the tubulin protein one. So microtubule go in length by adding tubulin. Dia boleh lebih panjang, panjang by adding the tubulin protein. So microtubule also can assemble and use tubulin to build microtubule. And then the two ends of microtubule are different to each other due to tubulin acts as dimer. One end can accumulate or release at much higher rate than other. This is important growing and shrinking during the cellular activity. So this one is that just mention that if you compare the two ends of the microtubule, it is totally different. It is are different to each other one. It look different one because tubulin is acts as dimer. You see why we call as dimer because it has alpha and beta. Tubulin is got two chains, alpha polypeptide chain and beta polypeptide chain. Okay, and then one end can accumulate or release at much higher rate than other. And so meaning that one end it more easily to receive the tubulin to form the microtubule, more longer microtubule or release the tubulin. Okay, so this is just a microtubule. And then what are the function of tubulin? So the function of tubulin, it forms the main structure cytoskeleton. So it gives the shape, provide the shape and mechanism support to the cell. And it divides the cytoplasm into the compartment to isolate the specialized enzyme. And then it contracts for the movement of cilia and flagella. So some of the cells contain cilia and flagella, right? So it provides the contract movement. So that the cilia and flagella can move, so that the cells can move. Not only muscle cell can be contract because of this. And also some cell that sperm cell can move because of this also. So it's very complicated, interesting, right? And then it also can pull chromosomes or chromatin during mitosis or meiosis process by central and serve as the thread for intracellular movement of organelles. Okay, so they are function of microtubule. So microtubule, one of the type of microtubule that found in the animal cell is centrosome. So you see this centrosome. So actually it is component of central. Ini central kan. So central actually it is one type of microtubule. Kalau banyak microtubule mereka combine, they combine together, they will form the central and centrosome. So when the microtubule go out, combine together, they will form centrosome that is located near the nucleus. And then the centrosome consists of a pair of centrols. Two centrols are arranged perpendicular to each other. So that's why you see the structure is like that. They are not arranged parallel to each other. They are perpendicular to each other. So key point. And then central is small and hollow cylinder shape because it is made up of microtubule. Right? And then central is composed of 9 sets of triplet microtubule arranged in a ring. Yang ni paling penting. It only 9 sets of triplet microtubule. So if you see this carefully see this structure, central is made up of 9 set triplet microtubule. So 9 plus 0. Why 9 plus 0? Because at the middle does not have any microtubule. Middle is hollow one, hollow thick one. Tak ada benda pun. So 9 set because of 1, 2, 3, 4, 5, 6, 7, 8, 9 sets. And then each set is made up of triplet microtubule, meaning each set contains 3 microtubules. So that is why we call as 9 sets of triplet microtubules. So this also the key points you have to know. And another one, why you need to know? Because of cilia and flagella. Cilia and flagella also the one type of microtubule also. So, but the structure is totally different from the central. So, if you see here, see this structure, see this structure. Actually, cilia and flagella of the structure of 9 plus 2. Why 9 plus 2? Because it is double microtubule only. It is not triple, double only. Dia hanya double je, double je. Dua only. And plus 2 meaning that there are 2 central microtubule at there. Dia tengah-tengah ada microtubule. Dua microtubule at there. So why 9 plus 2? So the differences is the outer surface got nine sets of microtubule, but each set of microtubule only have two microtubule only. And then at the tengah, at the middle part, it also have 2 microtubule. And then you will see this. Okay, inilah motoprotein that provide to the cell to move move. So that flagella and cilia will have many motoprotein at there, and then the microtubule also very packed together. Why? Because flagella and cilia is major component for the movement of the cells. Why they want to have the flagella and cilia? Because they need to move. Mereka nak bergerak. So if you see this, so this is the microscope structure. If you see the microtubule one, so if you besarkan dia, so dia akan macam ni. So ada 9 set kan, ada 2 tengah-tengah. And then yang ni pun sama. But there are 2 difference. Another one is basal body. Ini untuk cilium je. Ini untuk cilium. Kalau untuk basal body, it is 9 plus 0. So the 9 outer doublet of a cilium extended into the basal body. Actually, you see this. Actually, the this microtubule is extended into the basal body. Dia terus pergi ke basal body. But inside the basal body, each doublet join another microtubule to form a ring of 9 triplet. Each triplet is connected to the next by non-tubulin protein. So this is the 9 plus 0 chain arrangement. So dia tukar balik kepada 9 plus 0 arrangement like this. So please alert the difference. So kalau 9 plus 2 yang ni adalah yang tengah punya. Dekat. Memang dia yang dekat itu flagellum or cilia punya. Tengah-tengah punya yang ni punya. Yang ni adalah basal body, meaning it is already at the end point of the flagellum and cilia already. This one is the end point lah. Ini cell lah. Yang ni bawah ini semua cells. Yang ni adalah flagellum and cilia lah. Yang ni adalah endpoint already. What we call as basal body. But in basal body, they return back together 9 plus 0, 9 plus O. Okay. So just like that. So this one just cilia and flagella structure. So you see that 9 plus 2 right. And then if you go to basal body, you will change to the 9 plus 0. And then this one is the cilia and flagella. So mostly in animal cell, what cell have flagella is about sperm cell. So sperm cell will use the flagella to move to swim to the female vagina and then go to the fertilization process. This one you will learn in semester 2, chapter 4. And then also cilia. So normally our epithelium, all of the epithelium, some of the surface epithelium got cilia, right? Like even got cilia, right? So this cilia actually they look like this, and then it can have a back and forth motion. It provides the power stroke to move the dia macam ni, move the substance to move the substance. Okay, so that's all for microtubule. Okay, next we go to microfilament. Okay, microfilament is the structure is totally different from microtubule. So if within core filament, they look like helix structure. So microfilament are the thin solid rows, also called actin filament. Dia pun juga called actin filament lah. Why called actin filament? Because it made from actin protein, globular protein called actin. And then it is the two intertwined strands of actin. So they look like helical structure. Because they are twist together, twist structure. They are twist together. And then the microfilament can form structural network when certain protein bind along the side of microfilament to allow next filament extend as bulge. So normally this filament found at there in human is found in the human muscle. So microfilament will contain the actin and myosin filament. So in human muscle, we not only contain actin, but we also contain myosin. So this one you will learn more deeper in semester 2. Right. So actually, if you think that you enter semester 2, you will not learn about semester 1, you are wrong. In STPM biology, is more all are related together. So even though you don't want to learn in semester 1, but in semester 2, you also need to learn. Yeah, right. And okay, so the main function is to bear the tension or pulling force. And then the 3D network inside the plasma membrane also helps supporting the shape of the cell. And then the nutrient absorbing instantaneous cell of microfilament make up a core of microvilli. Our microvilli our microvilli in our intestine, small intestine, we know that small intestine can have microvilli to increase the surface area of absorption, right? So this microvilli is actually made from the microfilament. The basic component is microfilament. Okay, and then the microfilament and thick filament, myosin, interact to cause the contraction muscle of the cell. So our muscle can contract due to these two filaments. So if these two filaments is absent or cannot work, our muscle cannot contract already. No contract muscle meaning we cannot do anything already. Orang mati lah. Orang mati je baring dekat sana je. Tak boleh buat apa-apa lah. If no muscle contraction. And implant cell actin protein interaction contribute to cytoplasmic streaming, which is the circular flow of cytoplasm in the cell. So yang ni adalah awak akan belajar juga semester 2, transport topic chapter 2. Semua related ni. So memang semester 1 semua basic je. So you should learn lah. Okay, yang ni adalah small intestine structure in the cytoplasm. So if you see that microfilament is at here, right? Microfilament. So microfilament they combine together, so they will form the microfilament. And then within the microfilament they have the intermediate filament. So this one I will explain later. Interfilament filament is another type of cytoskeleton. So microfilament is one of the basic components to form the microfilament. So the nutrient exogenous intestinal cell of microfilament so either make up of core of microvilli have the finger-like projection. And then this structure, sarcoma, you will learn in semester 2. I just want you to know, have the image, what are the thick filament and actin filament only, thin element only. So actually, actin filament is we learn one type of microfilament. And another filament is we call as thick filament. Actually, these two filaments, they are work together, interact together to cause the contraction muscle of the cell. So these two filament will undergo one sliding filament hypothesis that you will learn in semester 2, chapter 3, muscle contraction. You will learn about how the muscle can be contracted. So for now, I just want you to show the structure about the thin filament and thick filament only. So this structure later you will see in semester 2. And in plant cell, yang ni pun semester 2 awak akan belajar. In plant cell, actin protein interaction contribute to cytoplasmic swimming, which is the circular flow of cytoplasmic in the cell. So yang ni lah. So kalau ada actin, so it can contribute to the cytoplasmic swimming. Ini pun awak akan belajar dekat semester 2 also. Okay, so finally last, intermediate filament. Okay, so intermediate filament is more thin compared to the microtubule and also microfilament. Intermediate filament is made by the fibrous protein, not globular protein. Fibrous protein called keratin, like keratin, and then they are coiled together. Dia macam tali lah. You will see that dia macam tali kan, coiled together into the cable tali. And then it has the long diameter compared to microfilament, but it is shorter. The length is more shorter to the microtubule. And then it is only found inside animal cells, especially in vertebrates. And then it is more permanent fixture than microfilament and microtubule. So permanent fixture is meaning that it can be retained longer, more longer than the microfilament and microtubule. And then they can be deassembled and reassembled in various parts of the cell. And even cell is died, it often persists. So meaning that these organelles, although the cell are died, but this intermediate filament will not die. It will persist at all. It can function. It also can function normal. Okay. So intermediate filament is like this. You see that if they compound, if only one monomer is like this, if dimer ada dua lah, tetramer, and then it will form the shape of beta tetramer, and then it can form the supercoil shape. So this one is more complex. So normally it's a major role in reinforcing the shape of the cell, so also provide the shape, and then fixing the position of the organelles in the cell. Yeah, it fixes the organelle cell. So it ensures that the organelles cannot move sesuka hati lah. Example, nucleus located in the cage made of intermediate filament is fixed in by bunch of the filament extended into the cytoplasm. And also, this one also synthesize nuclear lamina. And you still remember what is nuclear lamina? We already forget. You get again, nucleus. And then supporting the cell shape that help to carry out its specific function. So one of the example function is to support the instantaneous microvilli. So this one, why they have the intermediate filament is support the microfilament to support the microvillus one. Okay. Oh, ada lagi, belum habis. The last topic, differential centrifugation. Ada lagi, ini memang last topic lah. So actually you already learn all about the cells. Semua cells awak dah learn kan? You already learn all the organelles. So as the scientist, you are the scientist. So how do you isolate these all organelles? If you want to study only maybe nucleus, you only want to study nucleus, or you only want to study DNA only, you only want to study RNA, you only want to study ribosome, you only want to study Golgi apparatus. So how do you isolate that? I want a Golgi apparatus only, I don't want ribosome. All other component saya tak mau. Saya hanya mau a Golgi apparatus. So what are the techniques we can use? We can use differential centrifugation. So these are the process. These are the process that we can use in the differential centrifugation. So in general, senang. So actually, you will prepare the tissue, animal tissue cells, tissue cell sample tissue, either in animal or plant either. Okay, and then we homogenize. Homogenize using blender lah. Ni blender biasa kan. Ni blender we homogenize. Why we want to homogenize? We need to why we need to homogenize? Because we need to break the cells open. Kami kena buka kerana kami kat mouse cells tu. Kami nak cells component. We need to break the cell wall. Cell plasma membrane and cell wall. Kami kena break dia. We use homogenize, kami akan break down and then it will release all of the component. All of the component within the cell akan release out. Okay, and then the suspension is then filtered through muslin cool to separate the big fragment away from the small fragment. Okay, and then the particles are suspended in the liquid medium and placed in the centrifuge tube. So dia akan macam ni lah, placed in the centrifuge tube. And then it will undergo centrifugation. So what is mean by centrifugation? Centrifugation dia macam satu process to rotate the tips in very high speed. So normally the speed is 10,000 or 20,000 per minute. So 10,000 meaning that in 1 minute it can rotate 10,000 times. So why we need to rotate this? Because we need to obtain the platelet and supernatant. So what are these? So supernatant is the solution yang awak akan dapat dua layer. So dapat dua layer. So the upper layer is the supernatant, the liquid one lah. The lower one is the platelet. Platelet is the will be the solid particle. So normally this one we will do many types of centrifugation to isolate different types of organelles. So normally the first time you do the centrifugation, so the nucleus will be in the platelet. The largest size of the organelles will be in the platelet. So nucleus akan dekat platelet. Yang lain organelle, ribosome, Golgi apparatus, DR, lysosome, semua tu akan dekat platelet, akan dekat supernatant. So we already can. So from here, actually, when we discard, throw away the supernatant, we can. You want only the DNA. You can directly use this platelet. But if you don't want, you can do one more time of supernatant centrifugation, one more time using the supernatant. Not all these. You just transfer this supernatant to another tube and then do another centrifugation. So you do another centrifugation. For now, the platelet will contain the second largest organelles. Normally is mitochondria and chloroplast. Mitochondria and chloroplast will be retained at the platelet. And then, if you don't want mitochondria or organelle, you can do a centrifuge again, and then you will get about the Golgi apparatus, endoplasmic reticulum, lysosome. All of these you will obtain it. Okay, if you don't want also, you can do the centrifugation again to obtain the final one, which is very small structure, smallest organelles, ribosomes. Yang ni paling kecil lah. So you can do many types of centrifugation based on the aims, what you want lah, organelle. So that is what we call as differential centrifugation, because we do many types. We differentiate then based on the centrifugation. So based on many types of centrifugation, we can get the many different type of platelet. So these are the explanation. You can read yourself. So that's all for today. So thanks for watching. So today class actually is more delayed, already delayed 40 minutes. So any question? Okay, no question. So I'm sorry because this chapter actually is very, I can say it's very long. It's very long chapter. So sorry to say that I delay the class. So hopefully you guys can learn many things through this chapter. So that's all for today. So hopefully you guys can get the information knowledge from this chapter. So these two topics. So tomorrow we will next Monday I will proceed to the another last two topics, plant specialized tissue and also animal specialized tissue. So hopefully I can done. If not, if time is not enough, I will separate into 2 days because that two topics also have many things want to explain. So this chapter actually is very long, I know, but actually is you already learn in experience. So it's very easy you to learn, but you have to learn in expert manner lah. Okay, that's all for today. I don't want to talk much. So no question. Okay, so no question. So thanks for attending the class. So okay, that's all for today. Thank you. Bye. Have a good night. Oh, bye.